what will happen if meiosis does not occur in the life cycle of an living organism? Actually, when meiosis occur, then only the chromosome number will be maintained same from one generation to the other generation. If such meiosis is not taking place, the gametes which are formed will be having the same ploidy as like that of the parent organism, and when the gametes are fused from two different parents or from the same parent also, so the daughter generation which have been formed will contain double the number of chromosomes. So when the gametes of that generation will fuse, that will form into the second generation of daughters, where they will have 4 times the chromosome number of their daughter of their grandparents. Likewise, the chromosome number will become double from one generation to the other generation. That is the reason why at least for at any stage of the life cycle, the meiosis should occur in the life cycle of a plants or the animals. So if meiosis does not occur, some unimaginable characters may incorporate into the organisms. So it is not possible to control the characteristic features of that organism, so that may lead to destruction of the universe also. So that is the reason why at one stage or the other stage of the life cycle, definitely the meiosis should occur in the life of a plant or a animal. Okay. Now let us come to the actual process of meiosis. So here in mitosis, the nucleus will divide only once, but here in meiosis the nucleus will divide twice, but the replication of DNA will occur only one time. It will not occur twice, but only one time the DNA will undergo replication, because here also the interphase will take place. Interphase will occur once in the cell cycle of meiosis. First the interphase will occur, then meiosis 1, then meiosis 2 will take place. Actually there is a small gap between the meiosis 1 and meiosis 2, that is called as interkinesis. So what is the difference between the interkinesis and the interphase means? During interphase we know that the DNA replication is taking place or DNA duplication is taking place, but during interkinesis it is completely a resting stage where there is no division of the chromosomes, there is no duplication of the DNA, nothing is taking place. So it is called as interkinesis, which is different from the interphase of the cell cycle. So the interphase of meiosis is same as like that of interphase of mitosis, where G1, S and G2 phases are taking place. So now let us start with the first, the meiosis 1. Meiosis 1, this is a stage which contains 4 substages as like that of normal mitotic phase, but here those are named as prophase 1, metaphase 1, anaphase 1 and telophase 1. In the same way, when it comes to meiosis 2, there also the same 4 stages are present, but those are called as prophase 2, metaphase 2, anaphase 2 and the telophase 2. So before that, let's start with the first stage of meiosis 1, that is prophase 1. Out of the meiosis 1 and meiosis 2, meiosis 1 is actually called as heterotypic division. It is actually called as heterotypic division or actual reduction division, we can call it as. Why? Because the chromosome number is reduced to half during meiosis 1 only, and whatever the daughter nuclei which are formed, daughter cells which have been formed because of meiosis 1, those two are different from each other. The chromosome constituent part are also different from each other. That's why it is called as a heterotypic division. So now let us start with the first stage, that is prophase 1. Prophase 1 is the longest phase of all the phases of meiosis. Sometimes it may take 1 day or sometimes more than 1 day also, depending upon the cell nature and the environmental conditions. So here during this prophase 1, so many biochemical changes or many significant changes are taking place. So those changes are studied under 5 substages. So the 5 substages of prophase 1 are leptotene, zygotene, pachytene, diplotene, and diakinesis. So the changes, the events of prophase 1 are divided into 5 substages. So those are very crucial and very significant for the entire process of meiosis. In the meiosis 1, the first stage, prophase 1. In prophase 1, let's start with leptotin. In the leptotin stage, the size of the nucleus will gradually little bit increases, and here the homologous chromosomes are present. Homologous chromosomes are those chromosomes which are similar in their size, shape and the genes which are present on them, but they are coming from the two different sources. So out of that, one is coming from the male parent, so we call it as a paternal chromosome, and other one is coming from the female parent, that is called as maternal chromosome. So here this maternal paternal chromosome will come near to each other, and in the animal cells they appear to be like a bouquet. So we call they form into a special structure called as bouquet stage in the animal cells. Here each of the homologous chromosome which are similar in their size, shape and structure, they come near to each other and they will form into a pairing like organization, but they does not fuse at any point. Such congregation or the coming nearing of the homologous chromosomes will be taking place during the leptotin stage, and here there is a plate like structure, and the nucleolar organization can be observed here in the leptotin stage. Then the second stage is called as zygotin stage. So in the zygotin stage, the synaptonymal complex, a protein complex called a synaptonymal complex will be formed. So if you see the structure of packaging first, one homologous chromosome, that is maternal chromosome and the paternal chromosome. Likewise, we can see the two structures which are near to each other. This type of organization can be observed during the leptotin stage. Nucleolus will be present. So this is a nuclear membrane. So this is a cell wall, cytoplasm, nuclear membrane, these are the homologous chromosomes. This is a nucleolus. During zygotin stage, the chromosomes, the special proteins will be formed. We call it as synaptonymal complex. This synaptonymal complex, which contains the tripartite structure, which is having one central and two lateral elements. One central element and two lateral elements are present. And these lateral elements are connected with the further lateral elements. So here if you see the structure, this is a recombination nodule which is formed during the next stage, which contains a recombinase enzyme. So this is a central element and these are the lateral elements. which are connected with the lateral elements like this. So this is a central element, one central element from here to here, and this is the lateral elements. On either side we can see the chromatids. This entire structure which contains recombination nodule along with the synaptonymal complex. So that is the structure related to ribonucleoproteins which are present in the synaptonymal complex. So here the pairing of homologous chromosomes will be taking place. The pairing of homologous chromosomes is called as synapsis. So here that is because of a synaptonymal complex which is formed there. So here the synapsis or the pairing will occur by three methods. One is called as procentric, pro-terminal, and intermediate. Procentric means the synapsis will start at the center and it will move towards opposite sides or opposite poles. And pro-terminal. So the synapsis will start at one end of the chromosome and it will extend towards opposite end. So here in the procentric we can say bidirectional will be present, but here pro-terminal unidirectional is present. Intermediate, at many points the synapsis will start and it will be taking place in the multiple directions. Likewise, the pairing will be taking place in the procentric, pro-terminal and the intermediate form, and the synapsis will occur in zipper form, as like a zip, closing of the zip and opening of the zip. So zipper like fashion we call it as. So likewise the ribonucleoproteins and the synaptonymal complex which is formed will helping in the formation of the pairing of homologous chromosomes. So here because of this one bivalents are formed. Bivalents are formed by the two homologous chromosomes coming from one from the paternal, one from the maternal side. Next stage is packaging. Packaging is the most significant stage of the entire process of meiosis. So here during this phase the bivalents are converted into tetrads. So tetrads means each chromosome contains 4 chromatids in it. And out of these 4 chromatid combinations, so 2 are considered as a sister chromatids and the other 2 are considered as a non-sister chromatids. The non-sister chromatids. So here what are the sister chromatids and non-sister chromatids means, when the bivalents are formed or tetrads have been formed, the 2 chromatids which are formed from the same homologous chromosome are called as sister chromatids, and one of the chromatid from one homologous chromosome and one more chromatid from other homologous chromosome is called as a non-sister chromatid pair. Likewise the sister chromatids and non-sister chromatids are present in the homologous chromosomes. So here homologous chromosomes we can see the presence of recombination nodules. This recombination nodules contains enzyme complex called as recombinase enzyme complex. Recombinase is an enzyme complex which is present in the recombination nodule. This enzyme complex contains various types of enzymes. So like U protein, endonucleases, exonucleases. So such type of various types of enzymes are present. So we call it as enzyme complex. So how this enzyme complex is useful, and how the crossing over is taking place because of this enzyme complex, let's discuss by using some diagrammatic schematic diagrams. Consider this as the sister chromatid pair. This is one sister chromatid pair, and the non-sister, one more sister chromatid pair, this one. So because of the activity of endonuclease, nicking will take place. Nicking means at one point the breaking will take place. So because of the break, so this type of changes can be observed in both of them. So a nick is formed somewhere here, and on this side also nick will be formed. So we can see this type of structure, fragment like structure will be separated from that one. This is related to this homologous chromosome. Further, other homologous chromosome also the same type of nicking will be formed because of the same type of endonuclease which is useful for the separating the fragment. The U chromatin, the U protein, because of the U protein, the separation of the fragments or the chromatid pieces will be taking place. So let's continue the diagram here. So like this, the separation of the fragments can be seen, or the separation of the pieces of DNA will be seen like this. This is because of the activity of the U protein, the separation of chromatid pieces will be taking place, and because of the activity of R protein, the exchange of the pieces will be taking place. So that can be shown like this. Likewise, the exchange will be taking place between the non-sister chromatids. Because these two are the sister chromatids and these two are sister chromatids. One of the chromatid from here, one of the chromatid from here will be acting as a non-sister chromatids. So this type of exchange of the pieces will be taking place. So the continuation will be here. Where the fragments have been exchanged between the non-sister chromatids. Likewise, in this non-sister chromatids, the gene sequence have been changed, and in this one also the gene sequence have been changed. So these are the changes which are taking place during the packaging stage. So we call this process as crossing over. And the exchange of chromatid pieces between the non-sister chromatids, so that appears to be like X-shaped structure. So we can see the formation of X-shaped structures called as chiasmata. This one is called as chiasmata, and the exchange of fragments between the non-sister chromatids, that is called as a crossing over. So this entire process leading to the change in the gene sequence of the chromosomes. When the gene sequence is changed, so if the same process is taking place for n number of generations, so that leads to formation of a new species, that is leading to the evolution. So that is the reason why packaging is considered to be the most significant stage of the entire process of meiotic division. If the diagram related to packaging have to be seen individually, so this is a cell wall, this is a nuclear membrane, and the spindle fibers will start moving towards the poles. Here the non-sister chromatids and the sister chromatids, they form like this. They start forming a pairs at one point. This pairing is can be seen at one place or more than at one place. Gradually, after some time, we can see that exchange of the chromatid pieces have been taken place. Like this, we can see some X-shaped structures where this part of the fragment have been joined to this point to this one. Likewise, the exchange of chromatid pieces have been taken place. This is a diagrammatic representation of packaging with the chromosomes. So this forms the chiasmata. This forms a spindle, nucleolus, nucleoplasm, and this forms a bivalent or the tetrad, bivalent or the packaging. So packaging tetrad. So likewise, the exchange of chromatid pieces is taking place between the non-sister chromatids. So during the packaging stage, leading to the crossing over process. The next stage is diplotin. During diplotin stage, whatever the attractive forces which are present between the chromosomes will gradually decreases. So here the attraction decreases because of dissolution of the attraction between homologous chromosomes will decreases. Due to the dissolution of synaptonymal complex. So here, because of this one, the chromosomes will try to move apart from each other. First, the change can be observed at a point called as centromere. So both the chromosomes will move apart from each other from the point of centromere, but they remain attached at the chiasmata region. So the chromosomes appears to be like this. So consider, in order to differentiate, we draw this as a dark one. So this is the exchange portion. So the centromeres are moving towards opposite poles, but they remain attached at the chiasmata region. So because of this repulsion, so we can exactly or perfectly see the presence of X-shaped structure, that is chiasmata is clearly visible during the diplotin stage. So this is what the repulsion between the homologous chromosomes is taking place because of dissolution of the synaptonymal complex. Gradually, as the force of repulsion is increasing, so the centromeres will move apart from each other, but the homologous chromosomes will remain attached at the chiasmata portion only. So this is a stage where we can clearly see the structure of the chromosomes. So these are the changes that are taking place during diplotin stage. Then diakinesis. Diakinesis, where the repulsion between the homologous chromosomes will goes on increases. So because of that heavy repulsive forces, so the chiasmata will move towards the tip of the chromosome. So we call that process as terminalization. The moving of the chiasmata towards the tip is called as terminalization, so that we can see the chromosome structures like this. Only the chromosomes are attached only at the tip region because of more and more repulsive forces between the homologous chromosomes. So here we can see X-shaped structure, but here X-shaped structure is not present. So apart from this change, what happens is endoplasmic reticulum, the Golgi complex, nucleolus, nuclear membrane, all these structures will start gradually disappearing during the diakinesis stage. So by the completion of this diakinesis, the prophase 1 is over, so that all the bivalents, all these are bivalents. This is one bivalent which contains 4 chromatids, which is diploid in its nature. So this is 2n in condition. So this all the bivalents are released into the cytoplasm by the end of diakinesis. Then after prophase 1, the next stage is metaphase 1. Metaphase 1 is a stage where the spindle fiber formation is completed. So the spindle fibers are formed, those are bipolar spindles. Single bipolar spindle is formed. So here this is formed by the centrioles and centrosome in the animal cell, whereas it is formed by microtubules in the plant cell. That is the tubulin protein, myosin and actin proteins are present, apart from the microtubular structures. So here in the metaphasic cell structure, all the chromosomes, the spindle will be formed on the both the sides. So the same thing, that orientation of the chromosomes to the equatorial region will be taking place. So here what the thing is, the spindle fibers will come and attach to the centromeres, whereas the arms are present on the equatorial plate, whereas the centromere is towards the pole. So this is the equatorial plate. So this is a difference that we find between the metaphase of mitosis and the metaphase 1 of meiosis. In the metaphase of mitosis, we have seen that centromeres are present on the equatorial plate, but here the arms are present on the equatorial plate and the centromere is towards the poles, or it is facing away from the equatorial region. So here the continuous fibers, discontinuous fibers are also being formed. So the chromosomal fibers are useful for attachment of the spindle fibers to the kinetochore of the centromere, where all the chromosomes are oriented on the equatorial plate. So then anaphase 1. During anaphase 1, the spindle fibers will contract. When the spindle fibers will contract, what happens is wherever the chromosomes have been joined, that part will be separated out. So that separation of the genomes is taking place, because this total is a 2n in condition. So this part is n and this part is n. So the two n's have been separated from each other. That is what we call it as a separation of genome. Separation of the genomes is taking place. This process of separation of genomes is called as disjunction. The separation of genomes is called as disjunction or segregation. Here, whatever the genomes that are separated, that is not the daughter chromosome. That is not the daughter chromosome. So here if you see the structure, each one is 1 n. So likewise because of the contraction of the spindle fibers, the genomes are moving towards the opposite pole. Each genome contains 2 chromatids and 1 centromere. So this is a change which is taking place during the anaphase. Then telophase 1. During the telophase 1, what happens is both the chromosomes which are moving towards opposite poles, they organize so that the nuclear membrane will start forming reorganized. Nucleolus will be reorganized. Endoplasmic reticulum and the Golgi complex are also been reformed. So likewise we can see by the end of telophase 1, 2 daughter nuclei are produced. And here the 2 daughter nuclei which have been formed, these are not similar to each other because they contain the sequence of genes which are different from each other. Dissimilar type of nuclei have been formed in this process. And in this one, the actual reduction division is taking place during anaphase. Actual reduction division means the genomes have been separated during the anaphase 1. So these are the changes which are taking place during meiosis 1. By the end of the meiosis 1, 2 daughter nuclei have been formed which are quite different from the parental nucleus which is participating in the meiotic division.